think we're going to proceed to our next speaker. We have Marwa Mahmoud coming to us from Ain Shams University in Egypt. She'll be presenting LncRNA LINC00511 from newly diagnosed Egyptian breast cancer patients, a tale of a promising diagnostic molecular biomarker. All right, Marwa, whenever you're ready, you can share your screen. Awesome.
Assalamu alaikum. Good day, good morning, good afternoon, everyone. And thank you very much for organizing committee for this kind invitation to join this exciting international conference. I'm thrilled to be able to share some of our work from newly diagnosed Egyptian breast cancer patients' liquid biopsy by sharing a tale of a promising diagnostic molecular biomarker. First of all, allow me to share some numbers and statistics about breast cancer. Breast cancer became the most common cancer globally as of 2021, accounting for 12% of all new annual cancer cases worldwide, according to the WHO. The Global Cancer Observatory, which is an interactive web-based platform representing global cancer, states that the incidence rates are high in more developed countries, while rates in less developed countries are low but increasing. In Egypt, the estimated number of new cases represents 11.8% of total cancer cases, while the 5-year estimated prevalence rate for breast cancer in 2020 was 61,160 cases, or approximately 14.2% in Egypt. Through the time, the mortality rate exceeds 10% number of cases. 15,000 new cases are expected to increase in the next 40 years to reach approximately 37,000 cases. Although a rapidly growing number of treatment modalities have remarkably improved patient survival, approximately 20 to 30% of early-stage breast cancer cases will eventually experience recurrence and develop distant metastases. Therefore, unfortunately, by 2040, the rate is expected to rise to more than 7,500 cases, mostly because early breast cancer ordinarily lacks symptoms, and in this way, the disease is usually analyzed at a progressed stage with poor prognosis. Our hypothesis depends on the Sustainable Development Goals, which are a call for action by all countries under all aspects to promote prosperity while protecting the planet. Goal 3 is about ensuring healthy lives and promoting well-being at all ages. And since early detection is the best protection, extensive of non-invasive ways to screen that can diagnose breast cancer before appearance of the symptoms are obligatory. Individual biomarkers for breast cancer, which can provide more specific information predicting patients' prognosis, are quite necessary due to the molecular heterogeneity of breast cancer. To date, biomarkers such as molecular subtypes, according to the gene expression profiles that showed evidence of clinical utility and were recommended for use in practice, include carcinoembryonic antigen and CA15.3, estrogen and progesterone receptors, human epidermal growth factor receptor 2, BRCA1 and BRCA2 genes if inherited. Since the discovery of DNA at the end of the 19th century, RNA has been extensively studied. In the late 1950s, the central dogma of molecular biology was first devised. 30 years later, the first regulatory non-coding RNAs were initially identified, such as H19 and exist, but remained exceptions until the early 2000s. When the sequence of the human genome was published in 2001, which showed that only about 1.2% encodes protein, since then, regulatory long non-coding RNAs have been characterized in many species and were shown to be involved in processes such as development and pathologies, relieving a new layer of regulation in eukaryotic cells. Depending on location, sequence, morphology, and structure, they have a lot of different functions. They may act as scaffold molecules to regulate gene expression via a sampling chromatin modifying complexes at special loci, or as a decoy to promote or suppress gene expression. Long non-coding RNA may also act as microRNA sponge called competitive endogenous RNA to reverse microRNA suppression of its target genes. Now, let me tell you the story of our magnificent biomarker. Firstly and basically, bioinformatics was done to select a novel long non-coding RNA in breast cancer and detect its expression level. At 2019, the Heng et al. Performed a heat map of differentially expressed long non-coding RNAs in breast cancer tissues and adjacent normal tissues generated from RNA sequence data of the whole transcriptome, sequencing of 33 breast cancer specimens. After filtration and consultation, we selected LncRNA as the target in our study. In previous studies, LncRNA was reported to exert an oncogenic function in many cancers, such as non-small cell lung cancer, ovarian cancer, and glioma. However, the clinical role as well as the functional mechanism and tumorigenesis in breast cancer is still unknown. LncRNA, an intergenic non-coding RNA whose sequences do not overlap protein coding genes, is an oncogen with length of 2,265 base pair positioned at chromosome 17 on the short arm band 13 and composed of 5 exons. Mechanically, it was found to be positioned in the cytoplasm rather than the nucleus, which suggests that LncRNA exerts its downstream effects by post-transcriptional epigenetic regulation. Our case control study was conducted over a period of 15 months after obtaining approval from the Ethical Committee of Faculty of Pharmacy in Shams University and by the Ethical Committee of National Cancer Institute in New Cairo, and after all individuals had signed their written consent form. The study recruited 70 Egyptian patients, and the sample size was calculated based on previous study in 2012. Blood sampling, RNA extraction, cDNA preparation, and quantitative real-time PCR were done according to manufacturer's protocol. Next, in order, the association with clinical pathological parameters, positive predictive values, and negative predictive values was correlated with the expression levels. The median age at diagnosis is 1 decade younger in countries of Europe and North America. And more than half of the patients are premenopausal. Pathologic grading showed a low incidence of grade 1. Grades 2 and 3 tumors were 78.6% and 21.4% respectively. Majority of tumors are invasive duct subtype, and the profile of hormone receptors is positive for estrogen receptors and/or progesterone receptors in more than half of the cases. Most of the patients' molecular subtypes of luminal A or B-like. None of the patients received radiotherapy or chemotherapy. Jumping to the results, LncRNA is significantly overexpressed in breast cancer samples compared to control normal samples. Expression in the breast cancer patients correlated with histological grades being overexpressed more in advanced stages. It also showed that LncRNA was overexpressed in patients with positive estrogen receptor, likewise in positive progesterone receptors. Additionally, expression was markedly increased in the breast cancer patients with tumor size larger than 2 cm and significantly upregulated in breast cancer patients with lymph node metastasis. Eventually, the most association that received our interest was that LncRNA showed significant overexpression in early clinical stages, suggesting a promising tool for early detection and accordingly better prognosis. More than that, LncRNA showed superior area under the curve, confidence interval, sensitivities, and specificities over the classical tumor markers. Coming up was the computationally predicted downstreaming microRNAs to define target interaction mechanism. As shown, the bioinformatic methods which are currently used to predict paradigms suggested that the interaction of microRNAs with their targets depends on the sequence, evolutionary conservation. Such methods identified tens or hundreds of targets for each long non-coding RNA. However, the false positive rates were reported to be high. Therefore, further clinical investigations were obligatory. MIR-185 was chosen to further investigate the proposed interaction mechanism between long non-coding RNA and microRNA as competing endogenous RNA. Subsequently, another bioinformatic tool suggested a sponge interaction depending on base pair complementarity. Long non-coding RNA SNAP2 bioinformatic software detected an interaction between the LncRNA and MIR-185 with 5p end. showing 72.2 similarity, while impulse matcher bioinformatic tool showed that NIR185 with 3p end had superior similarity percentage of 73.7% at the position 525 on exon 5, which was previously confirmed by Liu et al. At 2018. So in conclusion, our team completed work on NIR185 3p. This figure focuses on long non-coding RNA blocking the negative regulation exerted by microRNAs. In figure A, microRNA block translation by binding to messenger RNA. While in figure B, long non-coding RNAs function as a sponge for microRNAs, so the messenger RNA translation is allowed. Our team lab will continue the tale by searching 5 possible suggested single nucleotide polymorphisms, depending on literature and a 2020 published article, as well as SNPs predicting bioinformatic tool. Eventually, the clinical pathological features will be correlated with the study terminating the tail, but initiating endless experimental opportunities for research. When all is said and done, I would like to express my profound gratitude to Professor Dr. Nadia Hamdi, my main supervisor, who started the proposing in this master project and believing in my capacity to accomplish it. I'm very grateful for her permanent support, scientific guidance, and encouragement throughout this long walk with all its ups and downs. I am also very grateful to Dr. Iman Sanat for her important considerations and suggestions in order to improve the biological activity studies, the scientific guidance, and for always pointing out the relevance of the results. I deeply thank my supervisor, Assistant Professor Dr. Inham, for providing all the necessary means for the clinical experiments, the scientific guidance into the field of pathology, the good advices, cheerful mood, and support whenever needed. And of course, the rest of the tail investigator groups, Dr. Dina, Professor Ahmed, and the sweet teaching assistant Shuru. Finally, I wish to express my love and my gratitude to my family for their constant support, encouragement, never-ending patience without whose love this work would never be possible. And thank you for your attention.
Thank you, Marwa. That was amazing.
Thank you.
We just have one question in the chat. Also saying thank you and asking if it is normal to have side effects like nausea, vomiting, or something more severe.
Nausea and vomiting?
I assume
Sorry, I'm not.
This wasn't for this talk. That was from an earlier talk, okay?
I haven't expressed any treatment options, so it's only diagnostic and prognostic tool.
All right. Well, we don't have any other questions. Oh.
I'll ask a quick question, Aliyah, if I may.
Sure.
So you mentioned this as a new diagnostic for breast cancer. And I'm just wondering, over the different stages of breast cancer, whether you anticipate that there might be changes that would have an impact on this as a diagnostic. The utility of it.
I think our population had quite covered all the stages possible. 70 patients are enough to cover, as the study sample size study estimation allowed only 25 samples. So we recruited 70 patients to cover up all the stages. So I think LncRNA showed good results in both stages, early and late.
Because as I mentioned earlier, I know in Kenya that people really only come in for diagnosis pretty late. They stage 3, stage 4. So an early diagnostic would be great.
Yes.
Pam, do you also have a question?
Yeah, but there are also some in the Q&A, so you could ask those if you'd like.
All right. We have a question asking, are LncRNA high levels specific for breast cancer?
I appreciate your concern, but it showed overexpression in many cancers as well. So it's not only specific for breast cancer, but it showed high specificity as well.
And then are there advantages of LncRNA over other breast cancer markers?
The main advantage is it detects the early breast cancer patients. I think that's the main concern about our study, early diagnosis.
Awesome. And then one more question from the chat. Is there a relation between LncRNA levels and response to the therapy?
I'm not sure what the research says on this. I don't have the data right now about the therapies. Our concern was only, our scope of the research was only for diagnosis and prognosis.
All right, thank you.
You're welcome.
Maybe I'll just also ask quickly is what I was really impressed about was the kind of, I suppose, especially in the patients with the bigger tumors, how accurate the LncRNA was in, acting as a biomarker. I'm wondering about the, I suppose, background of the patients. Are these all Egyptian patients? Have you tried to look at maybe some more global data sets to see if this is a marker that you have some regional specificity for, or if it's, because sometimes you do get this variabilities based on where the patients are coming from, right?
Yeah. Unfortunately, our patients were all Egyptian, but maybe in the future perspectives, we will recruit more global patients.
Really, really wonderful presentation, Marwa. Thank you so much.
Thank you. Thank you very much.
Yes, thank you again.
Thank you.